All right, welcome everyone. Today, we're going to take a tour through what I like to think of as a high-tech molecular factory. Our whole mission is to see how life's most essential work actually gets done, how the very reactions that build us and power us are happening millions of times a second. Okay, let's dive right in.
So, every factory floor has its challenges, right? Its bottlenecks. And in the world of biochemistry, there's one massive problem that slows everything down. The big question is, what is it? What's the single biggest barrier stopping a chemical reaction from just, well, happening? And the answer is something called activation energy. You know, the best way to wrap your head around this is to picture yourself trying to push a giant heavy rock over a hill. That first really difficult push you need to give it just to get it to the top, before it can roll down the other side, yeah, that's activation energy. It's this energy barrier that every single reaction has to overcome. And here's the thing. If that hill is really, really big, it takes a ton of effort and a ton of time to get that rock over it. In chemical terms, a huge activation energy means the reaction is incredibly slow. We're talking years, sometimes even centuries to happen on its own. And that's not exactly ideal for the split-second timing you need to, stay alive.
Okay, but what if the engineers in our little molecular factory came up with this brilliant breakthrough, a way to completely bypass that enormous hill? What if there was a shortcut?
And this really illustrates the solution perfectly. Instead of going through all the effort of pushing that rock over the massive hill, what if you could just build a tunnel straight through it? Your starting point and your ending point are exactly the same, but the journey? Suddenly, it's a whole lot easier and way, way faster. This is precisely what enzymes do.
So, to sum it all up, enzymes are what we call biological catalysts. They basically are that tunnel. It's super important to remember they don't add any extra energy. They're not pushing the rock any harder. They simply find a much more efficient path, lowering that activation energy hill, and because of that, they dramatically crank up the speed of the reaction.
So how do they actually do it? This is where it gets really cool. This mind map here, this is basically the blueprint for our entire factory tour. It's our guide to the 4 key strategies, or mechanisms, that enzymes use to create that tunnel. So let's take a closer look, shall we? All right, we're zooming in now. We are going right inside the enzyme into a really special spot called the active site. I want you to think of this as the main factory floor. And on this floor, there are 4 specialized stations, all working together to get the job done. Okay, our first stop is Station 1, proper alignment. See, for a reaction to even have a chance of happening, the molecules involved, we call them substrates, they have to collide in just the right way. And honestly, one of our sources put it perfectly. Imagine you're trying to connect 2 specific puzzle pieces, but you're blindfolded and they're just floating around randomly inside a big box. The chances of them bumping into each other in the perfect orientation are, well, incredibly slim. That's the challenge for molecules just floating around inside a cell. And this is where the enzyme's active site comes in. It acts like a perfect little jig or a clamp on the factory floor. It literally grabs the reacting molecules, brings them super close together, and then locks them into the exact perfect position for the reaction to happen. It just completely removes all that randomness, making the connection almost a sure thing. So the enzyme acts as a clamp, holding things in place, but it's actually doing a lot more than that. Let's move on to our second station to see how the environment itself plays a huge role. Yeah, that active site isn't just a clamp. It's more like a 5-star custom-built workshop. For example, the inside of a cell is mostly water, which can actually get in the way of some reactions. The active site can create a tiny water-free pocket. It can tweak the electrical charges, optimize the shape. Basically, it creates the absolute perfect conditions for that one specific reaction to happen as efficiently as possible.
Now we're at Station 3, and this is where the enzyme gets really hands-on. We call this bond manipulation.
I just love this analogy. If you want to snap a stick in half, what do you do first? You bend it, right? You put stress on it. It's so much easier to break once it's already strained. Well, enzymes do the exact same thing, but to chemical bonds. So here's what's actually happening. The enzyme physically grabs onto the molecule and literally bends or stretches it. This stress weakens the chemical bonds that are already there, making them much, much easier to break. And at the same time, it's bringing the right parts together, helping the new bonds to form. It's this really active physical process of breaking and making connections. And that brings us to our 4th and final station. And honestly, it's the most sophisticated one on our tour, transition state stabilization.
This is arguably the most critical job that an enzyme has.
Remember that rock on top of the hill? The transition state is that single, wobbly, fleeting moment right at the very peak, where the rock is perfectly balanced, just about to tip over. In chemical terms, it's this super unstable, high-energy moment where the old bonds are in the process of breaking and new ones are just starting to form. It's the highest point on our energy hill.
Now, this unstable state just wants to fall apart immediately, but the enzyme's active site, get this, it's perfectly designed to fit and bind to this wobbly transition state, even better than it fits the original molecules. It uses all these temporary interactions to kind of hold it together, almost like a scaffolding that stabilizes this really shaky structure. And by making the peak of the hill more stable, it effectively lowers its fight.
And with that, our tour of the factory floor is complete. We've seen all 4 stations at work, so now let's zoom back out and take a look at the final production report. What's the bottom line here?
And the data, it's just crystal clear. Without our enzyme factory, the energy barrier is huge, and the reaction speed is painfully slow. But with enzymes, the barrier is lowered and the reaction becomes massively faster. I'm talking millions, sometimes even trillions of times faster. The efficiency gains are they're staggering.
So why does any of this molecular factory tour stuff matter? Because these aren't just abstract ideas. These enzymes are the tiny microscopic engines running every single process in your body right this very second. And this, right here, is the ultimate takeaway. The enzyme doesn't change what you start with or what you end with. The rock still gets from one side of the hill to the other. But it makes it all happen on a timescale that makes life, well, possible. Without this unbelievable speed, everything, metabolism, DNA replication, you name it, would just grind to a halt.
So, this leaves us with one last kind of fascinating thought. We've seen just how perfectly designed these little machines are for building their tunnels and speeding up life. But if enzymes truly are life's perfect machines, what happens to the factory and to us when they break?